Okay, hi. So over here I have a functional group, and functional groups are made of hydrogen bonds, and hydrogen bonds are intermolecular, and they are interactions where the slightly positive H or one molecule is attracted to a slightly negatively charged atom of another molecule. And so the first functional group that we have is this one, which is a hydroxyl group, which is a O bonded to an H. The next one we have is a carbonyl group, which is C double bonded to O. Over here we have a carboxyl group, which is C double bonded to O and single bonded to OH. And if we come over here, we have an amino group, which is N bonded to 2H. This one is a phosphate group, which is phosphorus bonded to 4O. And lastly, we have a methyl group, which is C bonded to 3H. Okay, so over here is the plant cell, and this is the nucleus. And inside of the nucleus, we have the nucleolus, which plays a key role in the transcription and processing of RNA. And it's made of RNA and proteins, and it's also a cell organelle. The nucleus is the control center of the cell, and it controls and regulates activity in the cell. And an example of that is growth and metabolism. This over here is a vacuole, and a vacuole is used for storage, biomolecules, water, waste, etc. And it's usually large in plant cells but not in animal cells. And it carries water and other nutrient, and it is used as waste storage. This blue thing here is the mitochondria, which is the site of cellular respiration, which is the transfer of food energy to ATP molecules. The mitochondria has a smooth outer membrane but a highly folded inner membrane, and it is the site of cellular respiration, which is of glycolysis and the citric acid cycle and oxidative phosphorylation. Cellular respiration generates 2 ATP, and the overall equation for it is C6H12O6 plus O2 comes out to be CO2 plus H2O and ATP. This over here is the chloroplast, and this is the site of photosynthesis, which is the transfer of light energy to sugar molecules. And the chloroplast is double membrane, and it has stacks of thylakoids surrounded by stroma. And it's only found in plant cells and not in animal cells. And in photosynthesis, the equation is CO2 plus H2O plus light comes out to be O2 plus C6H12O6. And the steps of photosynthesis are light reactions and the Calvin cycle. Over here we have the Golgi apparatus, and the Golgi apparatus is a cell organelle that helps proteins package proteins and lipid molecules, and especially proteins that are planned to be exported from the cell. Over here is the endoplasmic reticulum, and the rough endoplasmic reticulum is actually covered in ribosomes, which makes proteins, and the smooth endoplasmic reticulum has no ribosomes, which makes lipids. So this yellow part right here is the plasma membrane, and the plasma membrane provides protection for the cell. And next to it is the cell wall. The cell wall is a tough, rigid barrier beyond the plasma membrane of the plant, and it provides support and mobility. In our animal cell over here is a lysosome, and the lysosome contains enzymes to break down old waste and old cell parts, etc. And it has vesicles with digestive enzymes that break down food molecules that are in animal cells, but they are not in plant cells. Okay, so here's a diagram to represent passive transport. And passive transport is high concentration to low concentration, and it does not require energy. So if we come over here and break this block, we can see how the high concentration of fish in this pool will come into this pool over here. Osmosis is the diffusion of water across a selectively permeable membrane, and osmoregulation is the control of water levels in body cells. So water actually follows salt. So hypertonic solutions are having less solute, while isotonic solutions have equal solute, and hypertonic solutions have more solute. And plants are constantly surrounded by hypotonic solutions. And active transport is the movement of molecules across a membrane that require energy and transport proteins, and that energy is provided by ATP. Active transport is low concentration to high concentration, which is also up and against the concentration gradient. ATP structure is adenosine triphosphate, which is the primary carrier of energy in cells, which provides energy for the physiological processes such as muscular contraction, and it works by adding its third phosphorus to other molecules. Energy coupling is using energy released by exergonic reactions to initiate endergonic reactions, and an endergonic reaction is something that requires energy input, and the reactants contain less energy than the products, and it is photosynthesis. An exergonic reaction produces energy output, and its reactants contain more energy than its products, and it's cellular respiration. Enzyme action can be known as lock and key action, and it's highly selective proteins with unique 3D configurations.
Hello, okay, welcome to part 2 of this video. Okay, so very quickly, let's go over the animal cell. So I guess we should just start here from the simplest area, the cytoplasm. The cytoplasm is responsible for holding the components of the cell and protects them from damage. It stores the molecules required for cellular processes and is also responsible for giving the cell its shape. Over here is this mustard looking thing is the Golgi apparatus. The Golgi apparatus is responsible for transporting, modifying, and packaging proteins and lipids into the vesicles for delivery to targeted destinations. Next up is the rough endoplasmic reticulum, which is this ketchup looking thing over here. So the rough endoplasmic reticulum plays an important role in synthesizing proteins destined for secretion from the cell. It also translates mRNA containing the code for these proteins. So this whole thing as a whole is the nucleus, and it basically controls and regulates the activities of the cell, for example, growth and metabolism, and it carries most of the genes and structures that contain the hereditary information in the cell. It's getting kind of rainy, let's fix that. There we go. Next up is the nucleolus. The nucleolus is concerned with producing and assembling the cell's ribosomes. Following assembly, ribosomes are transported to the cell cytoplasm, where they serve as the sites for protein synthesis. Next up are the centrioles. So this white thing is the centriole. Centrioles play a role in organizing microtubules that serve as the cell's skeletal system. They help determine the locations of the nucleus and other organelles within the cell. These purple thingies, ribosomes are important because they are responsible for protein synthesis. Now this cool patterned figure is actually the lysosome, but Haley already went over that, so I won't repeat it. Now over here, this fancy looking ketchup pattern is the smooth endoplasmic reticulum. The smooth ER is involved in the synthesis of lipids, including cholesterol and phospholipids, which are used in the production of new cellular membranes. Moving on to the powerhouse of the cell, the mitochondria. Now this blue thing and this other blue thing and another mitochondria over here is the mitochondria. They basically generate ATP, hence their nickname, powerhouse of the cell. Now, starting out with membrane structure and the phospholipid bilayer. Now, I made this makeshift phospholipid bilayer. So, as a whole, the phospholipid bilayer is similar to a gatekeeper. It's responsible for the plasma membrane's selective permeability. This is also in the plasma membrane. These are proteins, and this is the protein channel. These are the glycolipids, and I know this one looks the same, but this is a glycoprotein. These red things are the hydrophilic heads, and these cobblestone, really they're cobblestone walls that are just vertical. These are the hydrophilic tails. Hydrophilic means water loving, and hydrophobic means water hating. The transport proteins are found in the membrane itself. Each cell has a plasma membrane or a filter that helps regulate materials moving in and out of the cell. Each plasma membrane has different transport proteins embedded within it, which are used to help with this process. Each transport protein only allows a certain molecule to enter or exit the cell. As suggested by their name, channel or pore proteins open holes in the membrane of a cell. Channels or pores are typically designed so that only one specific substance can pass through. So there are different types of pumps. There is the podium-potassium pump. It is this pump that creates the ion gradient that allows neurons to fire. This pump begins with its sodium binding sites facing the inside of the cell. These sites attract sodium ions and hold onto them. The next one is the sodium glucose transport protein that uses secondary active transport to move glucose into the cells. They are active in the intestinal cells and kidney cells, both of which need to move glucose into the body system against its concentration gradient. The last one is the gated ion channels are passive transport proteins that open in full response to specific stimuli. You may be familiar with voltage-gated ion channels, but as those can cause our neurons to fire in response to input from other neurons. So there are also glycoproteins, which are molecules that comprise protein and carbohydrate chains that are involved in many physiological functions, including immunity. So chapter 6 is a lot about anaerobic respiration, aerobic respiration. So I will insert some pictures while it's raining. Aerobic respiration is in the mitochondria, and it is a chemical process in which oxygen is used to make energy from carbohydrates. Aerobic respiration requires O2, meaning it needs O2 for the process to take place. Anaerobic respiration is in the cytoplasm, and it's also more commonly known as fermentation. So there are steps to fermentation. There's the aerobic process that produces 2 ATP from the glycolysis, and it is important because it creates energy when there is a lack of oxygen. So anaerobic respiration does not need oxygen, unlike aerobic respiration. So there are steps to cellular respiration, and it takes place, yet again, in the mitochondria, the powerhouse of the cell. Glycolysis is the first step that produces pyruvate molecules. Those molecules are used in the citric acid cycle, in which the output is 2AP. It generates 2 ATP, The last step is oxidative phosphorylation, by which ATP is synthesized when electrons are transported from the energy precursors produced in the citric acid cycle. The inputs are NADH, FADH2, and O2, and ADP, and the output is NAD+, FAD+, H2O, and ATP. Hi there, it's Editor Lauren. So I realized I forgot to talk about the cellular respiration equation. So the equation is glucose plus oxygen, arrow, carbon dioxide plus water plus ATP, otherwise known as C6H12O6 plus 6O2, arrow, 6CO2 plus 6H2O plus ATP. Hopefully that helped out a little bit. This takes place along the inner mitochondrial membrane. Another thing to add on about glycolysis is that it's sugar splitting and it's in the cytoplasm and it does not require O2. I made this cell cycle diagram. So this is a lot about genetic material. So it includes interphase, which is G1 through G2. The M phase consists of prophase, which is the pink, metaphase, the blue, anaphase, the gray, telophase, the orange, and cytokinesis, which is the pink. So in the G1 phase, a lot of it is just basic cell growth and increase in the cytoplasm. If the cell passes, then it moves on to the S phase. If it fails, it just stays in the G1 phase. A cell that typically does not pass the G1 phase is a neuron. Hey people, it's editor Lauren here. So as I was editing the video, I literally realized that I did not talk about prophase, metaphase, nothing like that at all. So I'll quickly go over that with you guys. So the pink slice is prophase, and prophase is the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. And then the blue slice is metaphase, and metaphase is a stage in the cell cycle where all of the genetic material is condensing into chromosomes. So it's going from chromatids to chromosomes. And if you guys remember, like maybe a week or two ago, we did a preview about matching up like metaphase, prophase to like these diagrams. I feel like that's a really good connection to make if you're studying for that. And then the next phase is anaphase, and that is the stage of mitosis and meiosis in which the chromosomes move toward the poles of the spindle. And then there is telophase, a nuclear membrane that forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. And then the last stage is cytokinesis, which is basically just the splitting of the cell. It's just like, oh yeah, okay, let's seal a deal, let's split. So yeah, I hope that helped out a little bit instead of me just like completely forgetting to go over it. So yeah, I hope you guys keep on enjoying the video. The S phase is the duplication of chromosomes. If that cell passes, it progresses to the G2 phase. If it fails, it is brutally destructed. In the G2 phase, there is growth preparation for division, and if it passes, it moves on to the M phase. If it fails, it is also brutally destroyed. And then yet again, that is the image of the cell cycle diagram. Other helpful vocabulary that's needed throughout this process is chromatin, which is the unraveled package of chromosomes. During the prophase, chromatin basically gets coiled up into chromosomes. Think of kind of packing up and moving. You pack up all of your things and move rather than moving one book at a time into the trailer. Other fun facts about the genetic process is parent chromosomes split to become sister chromatids, and spindle fibers are what divide genetic material in a cell. So I hope this really helped one with like their knowledge or review, or I really hope that this is an accurate representation of everything that we went over. So a quick overview of everything. This is the phospholipid bilayer. This was the cell cycle, or sorry, not the cell, yeah, the cell cycle diagram, excuse me, the animal cell, and the plant cell, and the functional groups, the functional groups. This is everything that we built, and, oh, of course, there was this little thing that was filled with fish, however, I have no idea where they went. Yeah, so I guess let's end on a happy, sunny end. Weather clear. time set. So thank you for watching and listening, and I hope you have a great day. Bye.